Good evening, people. Good evening. Good evening, Komada. How are you, people? We'll go with the discussion. We had to take BioEve 24th paper. BioEve 24th paper. Take that BioEve 24th paper. We'll do the questions. We'll start with the first. Can you mark up to 5? Can you mark up to 5? First 5, can you mark? Ramai, can you mark first 5 MCQs? Right. First question. Silady correct statement regarding major components of cell membrane consists of 3 ester bonds. Major component 3 ester bonds. You know, people, major component of cell membrane. Right. Consists of 3 ester bonds. Major component is polypeptide, right, people? Polypeptide. 3 ester bonds. Doubtful, ne? Fatty acids are saturated. No. All, in addition, polar molecule or small charge molecule is always linked to the phosphate group. In addition, an additional polar molecule or small charge molecule is always linked. Always one fatty acid molecule is replaced by a phosphate group. One fatty acid is replaced by a phosphate group. Right. One fatty acid is replaced by a phosphate group. That is correct. Fatty acid replaced. Correct. Hydrophobic tails consist of 2 fatty acid molecules. Hydrophobic tails consist of 2 fatty acid, not fatty acid, ne? Hydrocarbon tails. Hydrophobic tails consist of 2 fatty acids. Hydrophobic tails are hydrocarbon chains, not the entire fatty acid part. So I'm taking 4th as the best. 4th as the best. 2 fatty acid molecules. Hydrophobic tails consist of 2 fatty acids. Now, the resource says, we know that hydrophobic tails are formed by fatty acids. but actually the tail is a hydrocarbon tail. Let me read that part. The two ends are the phospholipids. The typically additional polar molecule or small charge molecule, phosphate group, choline, composed of two fatty acids. They composed of two fatty acids and one phosphate group attached to one glycerol molecule. The phosphate group gives negative electric charge to the phospholipid, and typically an additional polar molecule or small charge molecule is also linked, not always, sorry, is also linked to phosphate part. The two ends of the phospholipid molecules different in behavior. The hydrocarbon tails are hydrophobic. Hydrocarbon tails are hydrophobic. Hydrocarbon tails are hydrophobic. While phosphate group and its attachment is hydrophilic. So not the entire fatty acid. So I'll go with the fourth one. Question number 2. What is the answer? The main difference between TEM and SCM. TEM and SCM. Mostly coated with gold, stained with heavy metals. Gold thing we took for SCM, ne? Wrong. Beam of electron pass through the thin specimen, beam of electron reflect from the surface of the specimen. More electrons may get displaced in the region. We'll take people second or third. Ne? Second or third, third is best. These two statements are okay. Both statements are all right. Ideal to observe surface view. Transmission, no. Other statements wrong. Ne? Question number 3. Question number 3. Difference of prokaryotes when they are compared with the eukaryote is prokaryotes. I think people, fourth one is better. Show nitrogen fixation. Fourth is better. Right? First is also correct. First is also correct. First or fourth, but fourth is better answer. Question number 4, we will select the correct statement regarding Golgi apparatus. Golgi apparatus. Inner and outer surface can be identified as trans and the cis respectively. Inner and outer surface can be identified as trans and cis. Inner and outer. Trans and cis, neme cis and trans, ne? Wrong. Trans phase is located near the endoplasmic reticulum. Trans phase, cis phase, neither cis phase. Cis and trans. Secretory vesicles are budded out from the trans phase. Okay. Produce lysozymes. Lysozyme kila even I read it as lysosome. Right? Lysozyme wrong. Neither. Collecting, modifying, packaging, and distribution of carbohydrate wrong. Right. So we'll take third one as the answer. Question number 5. Correct statement regarding extracellular matrix. Main components are proteins and carbohydrates. They're asking correct statement. Ne? Main components are proteins and carbohydrates. ECM, extracellular matrix. Glycoprotein, collagen, fibronectin. Those are the materials present. Ne? Fatty acids are hydrocarbon tails, right? Someone is asking a question like that. I think I gave the answer for that. We'll try to go and select the answers according to the resource people. That is what we have to argue. Right. Select the correct statement about the extracellular matrix. Main components are proteins and carbohydrates. Layer of proteoglycan secrete. Layer of proteoglycan doubtful. Ne? I can't increase the volume, students. I can't. I can't try anything today. Reason the computer stucks. Okay, I'll see what I can do next time, but not today. Stuck now. I can't do anything today. I don't know. It stuck. I think it's one of my browser stuck. Now we lose. Yo. Is it okay now? Is it okay now? Is there an improvement in the sound level? I don't know. I'll continue. Right. So a layer of proteoglycan layer ne wrong neither, not layer ne network or something ne. So layer of um, layer is wrong ne. So we'll take layer wrong. Collagen fibers are connected with the integral proteins of plasma membrane via fibers. Collagen fibers are connected to integral proteins of the plasma membrane via fibers. Integral protein of the plasma via fibers ne the fibronectin ne via fibers or fibronectin. Fibronectin or filaments. What is given? Filament. Or fiber, I think fibronectin ne given as fibronectin. Fibronectin. So fibers wrong. Then fibronectin again. And then most abundant glycoprotein in the ECM of the animal cell is collagen. Glycoprotein collagen, okay. I think I can settle with that. ECM of all animal cell, is there a problem with that? All animal cells, wrong, then most, most. Then again wrong. In influence the cell behavior by involving mechanical signaling, hurry. I think 5 is okay, 5 is okay. Right, moving to question number 6. Can you answer question number 6, people? 6 to 11, can you mark? Which of the following event in the prophase of mitosis? Following is an event in prophase. His spindle formed by accumulated microtubule complex. Chromosomal arms of non-sister chromatin are attached. Chromosome arms of non-sister chromatin are attached. Mitosis wrong, ne? Chromosome move towards opposite poles. The nuclear envelope fragments. Wrong, ne? Then accumulated microtubules include centrosomes, spindle, asta in all cells. Sixth one first. I think we can agree with that first. Question number 7, the correct statement about following diagram. Correct statement about following diagram. It can carry energy anywhere in the body, ne? Cell. Not body, ne? Cell. It can be produced in the living body within a short time period. Short time period. Living body, the cell, ne? Wrong. It is immobile, wrong, mobile. When it is hydrolyzed, free energy yield 35, wrong. During hydrolysis, release high amount of energy due to removal of terminal phosphate group. We'll accept it. We'll accept it as a best answer. Right? But if you carefully check the resource book says it like this. See if they understand this ATP. It says hydrolysis along with water. Most biological reactions use energy released during the breakdown of terminal phosphate bond. ATP can be produced within living cells within short period of time. Using ADP, inorganic phosphate and energy, production of ATP within the cell is called phosphorylation. During hydrolysis of ATP, ADP and inorganic phosphate are produced due to the removal of the terminal phosphate. As a result, very high energy is released. This is because the reaction of ATP and water. Right? So therefore, we'll take fifth one as a best answer here. Which of the following is a coenzyme? Biotin okay, FAD okay, NAD plus is okay, vitamin no. Second is the answer. Which of the following is correct regarding photosynthetic pigments? Substances which absorb light. Correct regarding photosynthetic, they are substances which absorb light. Substances which absorb light. Correct, neither. Chlorophyll absorb violet, blue and red light. Chlorophyll absorb violet, blue and red light. What do you say about it? Then chlorophylls and keratins are main type of chloroplast pigments. Yeah, resource book say visible light. That's correct. Oh, light keyword, visible light keyword, slightly different. What issue? Chlorophylls and keratins and main type of chloroplast pigments. Chlorophylls and keratins are main type of chloroplast pigments. Carotenoids, neither. Carotenoid should be ne. Xanthophyll also present. Next one is chlorophyll is a key light capturing pigment. Chlorophyll is a key light capturing pigment. Key light capturing pigment. It's chlorophyll A, right, people? Chlorophyll B is most effective for blue and red light. So what's the answer? Second is better, ne? We'll go with the second one as a better answer. First is also correct, but only thing is the word visible light is not given. So what is the non-visible light? What is the non-visible visible light? What are the other lights available other than the visible light? And what are the X-rays? Is it light? You take them as light or electromagnetic radiation? I think ultraviolet, IR radiation, infrared. Right. So you can take ultraviolet. Ne? So why the fifth answer is wrong? Chlorophyll B is most effective for blue and red light pneumonia, the chlorophyll A. Ne? So for 8, it should be FAD plus. FAD is not plus, ne? FAD. NAD plus, but not FAD. FAD plus is wrong. FAD, ne? Right? FAD is not FAD plus, ne? FAD. It says FAD plus. FAD plus is wrong. You check the nucleotide part, right? So it's not comes, it doesn't come as FAD plus. So some small errors are there in right? It should be FAD. No? Right. Right. Next one, you check in the nucleotide part, right? Check in the nucleotide part. Don't check in that photosynthesis part. Not don't check in the enzyme cofactor part. That is wrong. In that point, printing error present. You check in nucleotide part. Okay. The next one. Next one, question number 10. So I'll go with the second one for 9. Question number 10, the use of proteins, carbohydrates and fats in cellular respiration. Carbohydrates enter into cellular respiration pathways at the glycolysis. Carbohydrates enter into the cellular respiration pathway at glycolysis. Then carbohydrates enter into enter into cellular respiration pathway at glycolysis. Carbohydrates enter into cellular respiration pathway at different stages. Amino acid directly enter into cell respiration? No, deamination. Amino acids into the cellular respiration pathway at glycolysis. Can't they enter cellular respiration pathway at glycolysis? And then fatty acids enter into cellular respiration pathway at glycolysis. Wrong, the fatty acid enter as acetyl coenzyme A. So we'll go with the first one. Carbohydrates enter as glycolysis. Next one, question number 11. Select the correct statement regarding plant tissue system. Correct statement regarding plant tissue system. Visible elements and tracheids are present in all vascular plants. Sorry, vessel elements and tracheids. All vascular plants, wrong. Sclerites are found in places where growth is stopped. Sclerites growth stop. How about that? Okay, need. Sclerites correct, ne? Then cell walls of collenchyma are unevenly thickened by lignin. Cell walls of collenchyma are unevenly thickened by lignin. Not lignin, ne? Cellulose. C tube elements are present in some gymnosperms. No. Vessel elements are available. End walls of the C tube elements are completely broken down to allow movement of fluid. C tube elements, ne? Vessel elements, no. So we have the answer now. Sakar. Right. Question number 12, 13, 14. We'll mark them too. Select the correct statement regarding secondary growth of plants. Resins and other compounds permeate the cell walls of the wood. Cell walls, the resins and other compounds are permeated the cell cavities of wood. Cell cavities of wood. They are asking correct only. Cell cavities of wood. I think cell wall of wood, ne? Cell wall should be cell wall, ne? Cell wall. Next one. Check that statement. Well, I'm checking it at the moment. It says that the heartwood, generally darker sapwood, and other compounds permeate the cell cavities and help to protect the core of the tree from fungi. The heartwood is generally darker. Not the wood, normal wood, heartwood. Not wood, heartwood. Right? It should be heartwood. So therefore, first statement is wrong. Bark include the vascular cambium and all tissues outer to it. No, vascular cambium exclude all tissues outside the vascular cambium. Cork cambium is formed by the inner layer of the pericycle of the dicot root. Cork cambium is formed by the inner layer of the pericycle of the dicot root. Cork cambium is formed by the inner layer of the pericycle of the dicot root. Inner the outer layer, the outer layer. Cork cambium and the vascular cambium both are formed by the outer layer. Lumen of the vessels are formed in period of warm and wet seasons are larger. Warm and wet seasons are larger. ඒක නම් okay වගේනේ. All cells in the periderm are densely packed. No, there are loosely packed ones are available too. These loosely packed ones are forming what you call the lenticels. So question 12, I can go with the fourth one. Question number 13, people, select the correct statement regarding 3 routes of radial transport. 3 routes. Active transport involves in transport of solutes from symplast to apoplast. Symplast to apoplast, wrong, ne? Active transport occur into the symplast, no. In symplastic route, water and solutes never cross the plasma membrane. Water and solutes never cross the plasma membrane. Wrong, ne? It cross at least once. Endodermis prevents solutes that having accumulated in the xylem from leaking back into the soil solution. Prevent solutes that have accumulated in the xylem leaking back, correct? One substance can use one route, no. Most of the water transport through plasmodesmata, no. Third is a good answer. That is a good answer. Question number 14, in ascent of xylem sap, addition of water molecules transmit the pull, no. Wrong, ne? The cohesion, ne? Energy consumed, no. Next one, xylem sap is transported in the xylem by diffusion. Xylem diffusion, no. Bulk flow, ne? Continuous water column is formed only within the xylem vessels. Continuous water column is formed only within the xylem vessels. Water potential of mesophyll cell is reduced. Not only within the xylem vessel, ne? Apoplastic pathway, neither. Continuous water column present. So I think we can go with the fifth one. Question number 15, in phloem translocation, water potential inside the C-tube element increase at the source. Water potential inside the C-tube element increase at the source. Water potential inside the C-tube element increase at the source. Inside the C-tube element increase at the source. Water potential inside the C-tube element increase at the source. Decreases, neither wrong. Diffusion is not involved. Diffusion is not involved. It's a bulk flow, right, people? Phloem transport is a bulk flow, right? Then phloem sap is pulled through the C-tubes. Phloem sap is pulled through the C-tubes. Nah, neither wrong, ne? The pressure flow, ne? Wrong. Neighboring C-tubes may carry sap in opposite direction. Hurry. Phloem unloading occur passively. Unloading occur passively, yes. Now the second statement, diffusion not involved. We know the overall phloem transport is an active process. But when concentration difference is present, diffusion can occur. Now, you know, osmosis of what occur, right? So we'll take people 4 and 5. 4 and 5. Question 15, 4 and 5 as good answers. Second one actually doubtful, the validity of the statement. Question number 16. 16, during the closure of stomata, you have to select the correct response, correct order. Can you all mark the answers? During the closure of the stomata, curvature of the inner wall of the guard cell decrease. Curvature of the inner wall of the guard cells. Closure of the stomata. Curvature of the inner wall of the guard cells decrease. Correct, Nathan. Curvature reduce, inner wall. Exosmosis take place in neighboring cells. Correct. Potassium ions accumulate in guard cells. Now, exosmosis take place in neighboring epidermal cells. Wrong, Nathan. Exosmosis, this is wrong. Why? Exosmosis take place in neighboring epidermal cells. No. No. During the stomatal closure, water enter from the guard cell into the epidermal cells. So, endosmosis occur into the neighboring epidermal cells from the guard cell. Wrong, ne? Potassium ions accumulate in guard cells. Wrong. Turgor decrease in the guard cell. Correct. So, A and D. Third. Question number 17. When vacuolated cell is placed in pure water. When a vacuolated cell is placed in pure water, pressure potential gradient becomes 0. No, vacuolated means cell with a vacuole immersed in pure water. Water go into the cell, because cell always has some solutes in the cytoplasm. So water go in means the cell become turgid. So pressure potential gradually becomes 0. Wrong. Protoplast shrinks. Wrong. Protoplast expand. Turgor pressure exert on protoplast. Correct. Psi S and psi P values become equal. Correct. Pure water. So water potential becomes 0. So psi S and psi P values become equal and opposite in sign. So I'm taking the answer 17th question. C and D kilogam. Third. Question number 18. Characteristic features. Characteristic features shared by both Seacus and coconut tree is presence of compound leaves. Correct. Presence of triploid endosperm. Wrong. Seacus has haploid. Coconut has tree. N. Formation of archegonia. Wrong. Seacus has coconut tree. No archegonia. Flowering plants don't have archegonia. Then absence of secondary growth. No, Seacus is showing secondary growth. Wrong. Not released the megaspore to the environment. Correct. Both are same. So I'm taking A and E. 5. 18th question 5. A and E only. And next one is people 19. 19th question. Select the correct statement. Unisexuality is a typical characteristic of flowering. Unisexuality is a typical characteristic of flowers for cross-pollination. Unisexuality is a typical characteristic of flowers for cross-pollination. Is that correct? Unisexuality is a typical characteristic of flowers for cross-pollination. Correct. Gametophyte of angiosperm is reduced and structured with the 8 cells. I have to bring this to you when you come to the class on Saturday, okay? Gametophyte is of angiosperm is reduced structure with 8 cells. Wrong, 7 cells. Parthenogenesis cannot be induced by plant growth substances. Parthenogenesis cannot be induced by plant growth substances. Parthenogenesis cannot be, no, can neither. Wrong. Cannot be induced is wrong then. Then double fertilization, one sperm nucleus fused with two cyanogens. No. Seed consists of embryo and endosperm. Seed has embryo and endosperm. Seed has embryo and endosperm. What do you say about that? There's a seed coat also, ne? So doubtful. First one, unisexual is a typical characteristic of flower for cross-pollination. First is better. Is it correct? There's a small issue neither. Typical के word date पड़ी issue करती है आगे. Unisexuality is a typical characteristic. The special adaptation. Resource book says special. Special adaptation. You can't take typical. And typical means what? Present in all. So resource is special adaptation. I'll take that as a wrong statement then. Then the best answer, the closest answer is 5. Question number 20, people select the correct response regarding plant hormone and its function. Plant hormone and its functions. Cytokinin seed germination, okay. Ne? Gibberellin delay seed germination, wrong. Auxin promote leaf abscission, no. Ethylene delay senescence, no. Abscisic acid promote growth, no. First one. Question number 21, select the correct statement regarding chemical digestion. Small polypeptides are converted to small polypeptides convert to small peptides. Pancreatic carboxylase. Small polypeptides are converted to pancreatic carboxylase. Amino acids also, ne? Not sure. Second one, intestinal disaccharides catalyze the conversion of sucrose into monosaccharides. Correct. Fats are converted to triglycerides by the catalytic action of pancreatic lipase. Fats are converted to triglyceride by the catalytic action of pancreatic lipase. Wrong, ne? The fats are converted to triglyceride. Kill again. When wrong? Fatty acid, glycerol, and monoglyceride. Catalytic action of the intestinal nucleus convert DNA to deoxyribonucleotides. Catalytic action of intestinal nucleus convert DNA to deoxyribonucleotides. Catalytic action of intestinal nucleus. Intestinal nucleus wrong nail am I? Intestinal nucleus convert DNA to deoxyribonucleotides. This is done by pancreatic. Pancreatic carboxylase. Pancreatic carboxylase catalyze the conversion of polysaccharides into disaccharides. Wrong nila may heme carboxylase so linge hema karana carbohydrate digestion is wrong. So second is the answer. Name two hydrophobic molecules present in plasma membrane. Question is not very correct, people. Anyway, you can take phospholipids, glycolipids, and then cholesterol, hydrocarbon chains. So that question is wrong. I'm moving to next part. I'll skip that. Yeah, my computer stucks, people, today. Frequently it stucks. So I can't remove this chat now. Can't even. Right. What are main features of plasmodesmata? What are main features of plasmodesmata? What are you going to write, people? They are microscopic channels. We see that they are microscopic channels which run through the plant cell wall. Microscopic. You take the answer, but I can't clear this. I have a chat box open here. What are main features of plasmodesmata? Microscopic channels which run through the plant cell wall. They are cytoplasmic living connections. Right? They are cytoplasmic living connections. So the features of plasmodesmata, they are cytoplasmic living connections which run through plant cell walls. Next one, write two differences between peroxisomes and glycosomes. What are you going to write, people? Peroxisomes and glycosomes differences. Peroxisomes are present in plants and animals both. Glycosomes present only in animals. Sorry, plants. Glycosomes only in plants. Peroxisome, plants and animals both. Then you can see that peroxisomes contain enzymes to break down hydrogen peroxide. Glycosomes are having enzymes to convert fatty acids into sugars. Fatty acids into sugars. So you write like that, okay? I can't even close now. If I close, then I have to shut down the machine. So I'll go like this. Next one, write two differences between anapase 1 of meiosis and anapase 2 of mitosis. Anapase 1 of meiosis, you can see no sister chromatid separation, homologous chromosomes separate. Then anapase of mitosis, sister chromatid separate. Then you can see that the microtubule is attached to either side of the kinetochore in mitosis anapase, but they are attached to chromosomes. They are attaching the one pole attached to wait. We'll attachment we have to take for the metaphase. Ne? So what is the second statement we can mention, people? First answer second they're asking the masses 1 and anapase differences. First one we'll take that, people. Sister chromosomes are going to separate. Homologous chromosomes are segregating. Right? One difference. Then in anapase 1, homologous separation of one of you say that chromosomes attached at the centromere in masses. Wrong. Right? They are asking two. Anapase of masses. Now we can say centromere splitting, but it's the same point, ne? Centromere splitting, no centromere splitting. Anaphase 1. Anaphase. Kinetochrome microtubules get shortened, homologous pairs separate. One chromosome of each pair move towards opposite poles. Sister chromosomes of each chromosome remain attached. एक कन्फ्लूड नहीं. Sister chromosomes remain attached. Sister chromosomes separate. Right? Sister chromosomes remain attached. Sister chromosomes separate. But it's the same point. Closer points, नहीं? So we'll take it like that. Sister chromosomes. Right, next one is what is known as benign tumor. Abnormal cell. It is a tumor with abnormal cell lump. It is a abnormal cell lump remain at the original site. Abnormal cell lump remain at the original site. Abnormal cell lump remain at the original site. Next, we will, what are the three groups of lycophytes? Three groups of lycophytes. That's question is wrong, ne? Two groups of lycophytes. Or we'll say that two groups of lycophytes, two groups, ne? What do you call? Club mosses and spike mosses. We'll take it like that. Name two types of sporophylls in plants. The difference between them. Name two types of sporophylls in plants and state the differences between them. Microsporophylls and megasporophylls. Then microspores will produce microspores, produce megaspores. What is ascus? It is a sac-like structure formed by fusion of gametangia. Sac-like structure formed by fusion of gametangia. Gametangia during sexual reproduction. Gametangia during sexual reproduction. What are the two types of body forms, not five, forms seen in members of Cnidaria? Polyp and medusa. Polyps and medusae. Next one, write two differences between these two body forms. What are you going to write? Polyp attached, medusa free living. Polyp mouth turn up, medusa mouth turn down. Polyp tentacles around mouth. Polyp medusa tentacle around the margin of the body. I think you can write that. Next one, unique feature. Class Aves, keratinized feathers. Phylum Echondamata, water vascular system. Phylum Annelida, you can take Parapodia and Setae. Phylum Arthropoda, chitinous exoskeleton. Chitinous exoskeleton. Right? Next one, we will describe the structure of stoma. So, you have to describe, we learned the stoma structure. So, give details. They are epidermal cells, modified. Two types, bean shape and dumbbell shape guard cells are there. Stoma is formed by two guard cells. Wall thickness difference you have to mention, right? Those cellulose hoops, thickness difference, right? You have to mention they are specialized cells, chloroplast present, right? Those details, right? Then second part, describe the process of removal of water through hydrothodes. That is a good question. Hydrothode, how water is removed through the hydrothodes. We know people in some of the herbaceous plants, short growing plants, the in high humid night, what happened? They build up positive root pressure, and then as a result, the volume of water enter into the leaf is greater than the volume of water lost by the transpiration. As a result, through the margins of the tracheids, water and these the solutes can flow out. We call this as the guttation, right? So describe that process. I think there's a very nice the paragraph given in the resource book for that. So write the answer based on that. I'll give you a marking scheme, people. I can't do anything at the moment, right? The computer is stuck, so I'm going to finish it like this. Discuss major difference between the above mentioned process and transpiration. So difference between the guttation and transpiration you have to write. We learn people transpiration only water vapor is lost, and then guttation droplet, and no mineral loss in transpiration. Guttation mineral loss. Transpiration results what you call negative pressure in the xylem, and then it creates a transpiration pool. But on the other hand, the guttation occurred due to a positive pressure, right? So what about the CAM paper? I can't do that today, right? We'll take CAM paper for the next class. All right, I can't do anything. I have to shut down now. So likewise, we'll write the answer. I'll give you a marking scheme and I'll send it to the group. Okay. So thank you very much for staying this long with me. And then I'm going to say bye-bye to you. Everything is stuck. Okay. So then bye-bye. Right. I'm going to end the meeting.